My name is Daniel Peterson, and I'm a graduate student at UMass, University of Massachusetts Amherst, with Ben Normark. And today I'd like to talk to you about some work that we've done with Nate Hardy at Auburn on macroevolutionary trade-offs in plant-feeding insects. So plant-feeding insects are famous for being ecologically specialized. And in fact, a recent global survey of plant-feeding insects estimated that about 75% of species are restricted to host plants within a single plant family. Well, why is that the case? Why are insects so specialized when there seem to be so many obvious advantages to being generalist? You know, more food, more refuges from predators, that kind of thing. Well, one intuitive idea that's been around for a really long time is that being specialist is favored by natural selection because of trade-offs between alternative hosts. That is, an increase in performance on one host comes at the cost of decrease in performance on other hosts. And this is a model of how that might work genetically if you have a single genetic locus that affects performance on multiple host types. In this case, we have the blue host type and the red host type. And as you can see, the little a alleles in this model have a high fitness on the red host and a low fitness on the blue host, and vice versa with the big A alleles. And you can see there's a trade-off built in here because no genotype has the highest fitness across all host types. However, a lot of studies have looked for this kind of mechanism in wild populations of insects, and more often than not, they don't find this. So what's going on? Well, one possibility is that the trade-offs are actually acting over macroevolutionary scales instead of microevolutionary scales. If you imagine that the host-use adaptive landscape is rugged, with lots of peaks and valleys, which you might expect if there are, say, epistatic interactions between loci, that kind of thing, then it could be that specialization onto different host plants actually represent isolated fitness peaks in the landscape. And then what you have is a trade-off not between alleles at one locus, but between different evolutionary trajectories, of which a lineage can only take one. The problem is it's hard to find these because you're not going to be able to observe this within any given lineage. So how can you look for this in natural populations? Well, one thing you can do is look at trait distributions across phylogenies. If you think that trade-offs between hosts are going to be strong, you expect that performance on alternative hosts are going to be negatively correlated on a phylogeny. And here I've shown a couple different ways that could happen. If you take this to be an imaginary phylogeny of insect species, and those with red squares being those that have high performance on the red host, and with blue squares high performance on the blue host, you can see in both of these situations there's a strong negative correlation between performance on the two hosts. However, in the column A situation, you also have a strong phylogenetic signal, whereas you don't in column C. And that suggests that in the column A situation, you have some sort of long-term macroevolutionary process that's limiting transitions between hosts, which suggests that historical contingency might be involved in the trade-off between hosts that we're seeing here. However, in the column C situation, there's no phylogenetic signal, which suggests that it might be more of a macroevolutionary mechanism that's underlying this trade-off that we see. Of course, negative correlations aren't the only possibility. We could also imagine that there are positive correlations between performance on multiple hosts. And that positive correlation could happen over the phylogeny or over the short-term effects as well. You could even have, positive correlations on one time scale and negative correlations on another time scale. And any of these patterns would give valuable insight into how specialization is evolving in nature. And so in order to do this kind of analysis, you need two things. You need host-use information for a lot of species, and you need their phylogenetic relationships as well. And luckily for those of us that study plant-feeding insects, people have been collecting insects for a really long time, and it's also been common practice to write down what plant species they were collected from. And this data has been sitting in museums for a long time. And recently there's been an effort to digitize a lot of this information. And in particular for two groups, the Lepidoptera, the caterpillars, or the butterflies and moths, but I'm talking about their caterpillars and their host plants, and the Hemiptera, the true bugs. There are two really good databases online nowadays that have a lot of information for these two groups, and so that's what I'm going to talk about today. And we were able to get over 1,600 species for Lepidoptera and almost 1,000 species of Hemiptera, where we had host-use information and phylogenetic information. And so we decided to take this data and try and see whether we could find evidence for trade-offs within these groups. Now the trickier part is deciding how you're going to classify your host-use traits. Ideally, you would use host species because that gives you the most information and perhaps the most biologically relevant. Unfortunately, we found that we don't have a lot of statistical power to analyze host species because not many insects are on each one. So what we decided to do was aggregate them into host plant orders. And these 11 host plant orders ended up being the most commonly used for both caterpillars and bugs, which was interesting. And you can see it's quite a diverse group. of plants, but I want to particularly draw attention to the slide not changing. There we go. Okay, Pinales, which is our one gymnosperm group, the conifers, and Poales, the grasses, which are our one monocot group. And the rest of them are all dicot plant borders. And so we expected that we'd see the strongest trade-offs between these two and the rest of them, because it's the greatest phylogenetic distance. Okay, I'm just going to show you what the data look like here. Here's the phylogeny of the Lepidoptera, and here's our phylogeny of the Hemiptera. And then in rings around it, you have which of those species are present on which of the 11 host plant orders. So those with the green dots in the outer ring are the ones present on Asterales and so on. Now, it may be kind of hard to see the patterns from this scale, so I'm going to zoom in just a little bit. So this is just a segment of Lepidoptera, where you can see immediately that there's a good amount of phylogenetic conservation of host use. So this whole clade of Lepidoptera only uses the Poales and nothing else. And then this clade only uses the Pinales and nothing else. So you can see there's hints of negative correlations going on here. Now, if you look at the patterns in the Hemiptera, again, a non-random sample that I chose, you have a totally different pattern, right? You have much less specialization. Some of these insects even seem to be using all 11 host plant orders, which means they're extremely generalist. But we really wanted to go ahead and calculate quantitatively what is the relationship between the evolution of these different traits across the tree for both groups. So what we did was we constructed phylogenetic mixed models, and we compared each pairwise comparison between the 11 host orders individually. So there's 11 host orders, there are 55 pairwise comparisons that we did in our own model. And then for each one, we calculated the long-term phylogenetic correlation between the hosts, presence of the two hosts, and then as well as short-term residual correlation after accounting for the phylogenetic effects. And then we took all those pairwise correlations and we put them all together and made a big network to see how all the hosts relate to each other. And here's what it looks like for Lepidoptera. On the left, you have the long-term phylogenetic correlations, and on the right, you have the short-term residual correlations. And each of these dots represents a different host plant order. The size of the dot is proportional to the number of insects that are found on that plant order. And then the lines between them are the correlations, with black solid lines being positive correlations and red dashed lines being negative correlations. And then the thickness of the line is proportional to the strength of the correlation between the hosts. And then the hosts are also arranged spatially by an algorithm in order to maximize distance between hosts that are negatively correlated and minimize distance between hosts that are positively correlated. So to clump them spatially by those that they're positively correlated with. And in fact, if they form statistically significant clusters with positive correlations within the cluster and negative correlations between clusters, then those are outlined in gray. I'm not sure how well you guys can see those. But the most striking result is that in the long-term correlations within the caterpillar host use data, you have two significant clusters of hosts. And interestingly, those hosts are not seemingly divided by phylogenetic distances, because you have the dicots divided, and then the gymnosperms are in one group, and the monocots are in another group. It seems like they're more divided on ecological level, where this group of hosts are much woodier hosts, and these hosts are the more herbaceous hosts. We see a different pattern, however, when we look at the short-term correlations between hosts. You see in this case that all the angiosperms are in one cluster, and the Pinales, our lone gymnosperm, are isolated by themselves. So this appears more to reflect phylogenetic distance. And it's surprising because the phylogenetic distances between plants, the long-term diversification of plants, seems to influence the short-term evolution of the insect parasites. Whereas the short-term ecological differences between hosts appears to influence the long-term evolution of the insects, which we didn't expect. And then we see a totally different pattern again in the Hemiptera, the bugs. We see almost entirely positive correlations between hosts over both time scales, with all the hosts falling into a single cluster for both of them. And it's key to note here that these aren't just neutral correlations. They're not just an absence of trade-offs. These are strong positive correlations, especially over the short term, which suggests that adaptations to one host actually increase performance on average on all these other hosts, this very large diverse group of hosts. So what can we conclude? Well, number one, we do seem to find evidence for macroevolutionary trade-offs. However, it seems like those trade-offs vary over different timescales, long-term versus short-term evolution. We also find that these trade-offs aren't universal. there's no big law of herbivory that says you can't use pines and oaks at the same time. In Hemiptera, we see that, and so trade-offs apparently depend more on the historical contingency and genetic background of the insects that you're interested in. And finally, and this was a surprise to us, positive correlations between even these widely divergent host groups are very common, which suggests that generalist adaptations to multiple hosts are as important as, if not more important than trade-offs over the long-term evolution of host use in plant-feeding insects. And I guess I'd say the main takeaway I'd like you to get from this is that it seems like persistent, taxonomically broad trade-offs between different plant groups are not what's driving ecological specialization in plant-feeding insects. And given that microevolutionary trade-offs within plant-feeding insects populations have also been difficult to detect, it seems like the case that maybe neutral and non-adaptive processes are more important in the evolution of specialization than we've appreciated up to this point. Okay, let me just make a few acknowledgments. I owe a huge debt of gratitude to all the people and the hard work that went into making these host use data sets that we use for this analysis, including the Lepidoptera data, which is from the host database, that's from the Natural History Museum in London, and the Hemiptera data, which came from the Tritrophic database, which was an NSF-supported collaboration from a bunch of people, but it's now hosted at the American Museum of Natural History in New York. And with that, I'll take your questions. Thank you. Evan. Yeah, you might expect that, although I'd say it's not necessarily clear how diversification fits into this scheme. You could have, for instance, generalists evolving to be more generalized and producing specialist lineages from that then go extinct at some rate. So actually, that's something that we're very interested in pursuing in the future, is how diversification fits into this whole scheme, because it's definitely not straightforward. Although I agree that specialization has been one of the main reasons people think that plant-feeding insects are so diverse. And that could still be true, I'm not denying that. But definitely it's not as straightforward as people might think.
Kind of like along those ways, but provided that there is sampling issues, but some of the orders that you have in there are quite big.
The plant orders.
Yeah, so think about Australis, Physalis,
oh, yeah.
Have you tried to look at target families or suborders within those that may have the greater diversity and see?
Yes. Right, so if we may have missed a lot of trade-offs with this analysis, particularly chemical differences. And post-plant orders are huge groups. There's definitely a lot of them that aren't very well defined. And we did this sort of as a first attempt to see, with the largest groups that we could find, to get a statistical power, do we see the trade-offs that we'd expect? And we didn't. So definitely our next step is going to be trying to hone in more on traits that we would expect to produce trade-offs and see if we can find those. We might have 30 seconds. Nope.